Hello again, molecular ecology. Welcome to the main video for our anticipated results proposal work module. This module is a little bit shorter, so this is indeed the only main video in addition to the overview video. And in this particular video, I will cover some of the logistics, so what should be included in this section. And again, as I said in the overview video, this statement is really short, only about one sentence per aim. So that's also why this video is a little bit shorter than the previous ones, which was helping us build some longer sections of our proposal. So let's think first of all about the purpose of the anticipated results statement. So in this image here, I think what is trying to be shown is this idea of anticipating is thinking about what is going to come next. So as you're going through each one of these letters in the word anticipate, you're trying to think of what that next letter is going to be. So when you're anticipating something that hasn't happened yet. So when you're writing this proposal, you haven't done the study yet, right? So we don't know for sure what the results are going to be. But what we're trying to do with this anticipated results statement is have some kind of statement about what the potential results could be. And that's important because it helps convince the reviewers that you've thoroughly thought through your proposed work, you've done your homework, you've reviewed the literature, and so you've planned ahead. I liked this picture when I was Googling things making this lecture yesterday. This is an example of not planning ahead, right? If you're not leaving enough space in your line for that last letter. But the idea here is you are trying to convince the reviewers that you know what you're doing. And this is also actually something that if you don't have it listed, sometimes reviewers will ask for it. So not to say that this is a make or break, that if you don't include this, you're never going to get funded. But in fact, some of the proposals that one of my master's students has submitted, that was one of the main comments she got from reviewers, is that she didn't include enough information about what she expects to actually find in her results. So that's one of the reasons why I'm including it here. So what does this look like? Often what you will see these anticipated results statements look like is in some kind of format like this. Because of blank, I expect that blah, 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 or based on previous research, I expect that blah, blah, blah. And so again, really this is helping to show the reader that you're thinking like a scientist and you have the potential for success in your future career because you're able to formulate these hypotheses and ideas based on what you've been reading in the literature so far. So, and remember what we want to do is we have two aims, and so for aim 1 we will have, sorry, I accidentally stopped the recording. I don't know at what point it stopped, so I may repeat myself a little bit, but bear with me. So for aim 1, we have our stated aim, and then we have the approach section, which we covered previously about how we're going to do that experiment. And then this is something you would tack on at the very end of that approach section or paragraph, is one brief statement about what you expect your results to look like. And you would do the same thing for aim 2. You would have your approach section and then tack on a statement at the end that might say something like, based on previous research, I expect that blah, blah, blah. And so really what you're doing is thinking about, so when we're thinking about this part here, so because of blank or based on previous research, what you're doing is using information that you've already gained from reading previous studies, what is already known about the study organism. So a lot of this is stuff that you've already set up and background information you've already provided in the intro. So what you're doing is just building on that to provide one concrete sentence about what you think results you might expect and why. And so really what we're doing here is building off some of the information we provided in the approach section. So this is one of the slides from the approach section video previously, and this is one of the things I said that we should include in our approach section is the particular data you're going to obtain that will allow you to answer your research question. So in this example here, we might obtain data that gives us outlier loci. So remember, each one of these blue dots is a SNP. Any SNP in this red region here is an outlier locus under selection. So maybe the data you're getting is outlier loci, or maybe if you're doing something like a metabarcoding study where you're looking at taxonomic diversity of different bacteria, you might get a graph like this, where the information that you're getting is relative abundance or something like that. And so what you're doing then when you're writing your anticipated results is you're building off of that information you included in the approach and giving more specifics. So if we're talking about these outlier loci, you would specifically say what type of outliers you might expect to find in terms of what types of genes might be outliers based on previous studies that have been done. If you're talking about relative abundance, it might be what type of bacterial species do you expect to differ between your treatments, again, based on previous research. So we're building on information that we've already provided previously in the proposal, but we're providing some additional specifics. So not saying only I will determine which bacteria are different between groups X and Y, but now for anticipated results, we're saying I expect that this particular bacterial group will be different because of previous studies. So let's walk through an example of this, which is always helpful for me. This is an example from Dr. Caitlin Lauder's funded GRFP proposal. This is what we used in one of the quizzes previously for the approach, where I asked you to identify different sentences in her approach that hit the different criteria that we should be including. So again, this proposal was funded, she got a GRFP, and let's look here at what she did. So here she again is looking at spiny lobsters, and her aim 2 was to understand how different stressors affect gene expression. So that's what she's specifically interested in. So this is an excerpt from her approach section for her aim 2, and highlighted or bolded here in darker font is a sentence that says spiny lobsters will likely respond to multiple stressors by regulating genes like those that concentrate and bind calcium near the calcification site. So if I had to pick from her current proposal, I would say that this is her anticipated results. But I would actually recommend doing it a little bit differently. And again, this is important to note. There are slightly different ways to do it. The way that I'm telling you is not the only way to do it, but I think it makes sense based on some previous successful proposals I've seen and what I've been reviewing here for some proposals. So I suggest actually moving this statement to the end of her approach section. And so that might look something like this instead, where now here we have a similar sentence that's bolded at the end. So you start with your aim, you give your approach about how you're going to do the experiment, and then at the end you have that one sentence about what you anticipate your result to be. And I've modified it slightly, so let's read what it says. It says, based on previous research in crustaceans, I expect that spiny lobsters will respond to multiple stressors by regulating genes such as the sarcoendoplasmic reticular calcium pump that concentrate and bind calcium near the calcification site. So the naming of this particular gene is something that I actually added. This wasn't in her actual proposal. And this is just based on some Googling I did yesterday to find out what research they've done in crustaceans and what genes might be likely to be differentially expressed under these types of scenarios. So this is an example of this quick one-sentence statement you can put at the end of your approach section to give some idea of what you anticipate your results to look like. And so now you might be asking, okay, but how do I figure out what results I anticipate for my proposal? Am I just looking in a crystal ball and guessing? You probably know the answer is no. So I'll give you some tips here. And I think another important thing to point out is that obviously we want to make sure we're giving some anticipated results that are well informed and well thought out, but it doesn't matter if you end up being right or not. No reviewer is going to come back after you've done the experiment and say, hey, you thought that this was going to happen and that didn't happen. that's not really the point here. The point is really more to show that you've done your homework, you've thought through it, and based on previous research which you've reviewed, this is what you think is going to happen. So how do you do that? Some of this we've already done the legwork for. So what I suggest you do is find a reference paper or papers plural that investigates a similar study question as the one that you are proposing. And so this might be papers that you've already found because you've been doing a lot of research already related to your proposal. So you can start by looking at some of those same papers that you found for the intro or the background info, or papers you reviewed to see what type of approach to take if you're taking a similar approach as that type of paper. So maybe this is a paper you found when you were trying to figure out what type of approach to take. And when we were using the papers for approach references previously, we focus on the method section of that paper. But here, if we're trying to figure out what type of results we anticipate in our study, now we want to look at the results section. So this is a table that's an excerpt from the paper that's shown here. So in this paper, what I was doing was comparing individuals from different geographic regions. I knew that temperature was different across those different geographic sites, so I expect that temperature is one of the selective pressures causing differences. I identified outlier loci between those two different geographic regions, and this table here is listing the annotations. So remember, if you think back to the genomics and transcriptomics module videos, when you're annotating something, you're taking that unknown sequence of the gene, comparing it to a database to figure out what that gene name and function is. So here I'm showing there were, out of all the outliers that I identified, which ones I was actually able to determine the annotation for them. And remember, you usually cannot find the annotation for all outliers in a study like a dd-rad study like I did, because with genomic sequencing, you are looking at both coding and non-coding regions. So any outliers you identify in non-coding regions are not going to have gene annotations because they don't correspond to actual expressed genes. Okay, so in this table, it's showing us some examples of outlier genes that I found in my study. So these are just different gene names: dipeptidyl peptidase 6, NADH dehydrogenase subunit 1, etc., etc. So if you were also doing a study looking at different geographic populations of a species, and you knew that thermal differences, temperature differences were different across these different geographic sites and might be providing selective pressure for those populations, and you were identifying outlier loci in your study as well, you might say that you expect to find some of these genes that are listed here as outliers because it's been found previously in populations that are under different temperature stresses. So that's what I mean by using previous literature to inform what you expect to find in your study for your anticipated results. So just to recap, overall the different proposal components we've discussed so far. So for proposal 1, you covered the title, intro, and research idea and specific aims. In the last video, we covered the approach, and now we've tacked on that anticipated result statement. And so really now you have all of the information you need to write proposal 2. So our second proposal is going to contain all of these aspects plus the references. And then once we write proposal 2, we just have a few more sections at the end to tack on that will lead us to that full 2-page GRFP proposal. So we're getting there, you're making good progress, and now what you do is go through the Canvas module. There's one quiz that will ask you to identify the anticipated result statement from a provided proposal excerpt, and then you will also rate your own drafts of your own based on your own proposal, and we'll get you some peer feedback like we have been doing as well.